Hello and welcome to Pandemic Live. I'm your host, Russ Altman, and today I'm talking with Jean-Laurent Casanova of Rockefeller University about the genetic basis of the vulnerability to infectious diseases, including COVID-19, which I'm sure is on all of our minds. First, a few comments. I'm a professor of bioengineering, genetics, medicine, and biomedical data science at Stanford University. My research interest is in the application of computing, AI, and data science to problems in medicine. I'm also, and I think this is why I'm here today, the editor of the annual review of biomedical data science. And this is brought to you in part and entirety by the annual reviews organization. I'm the host of a radio show called The Future of Everything, which is also available on an iTunes podcast, different from the Wall Street Journal podcast with a similar name, unfortunate. This discussion is part of the Pandemic Life, an initiative to disseminate scientific ideas and findings that shed light on the COVID-19 pandemic. Pandemic Life is produced by the nonprofit publisher Annual Reviews as part of their commitment to bringing the knowledge and wisdom of the world's leading researchers to policymakers, practitioners, patients, and the wide general public. Now, over the last 25 years, Jean-Laurent Casanova has explored the genetic basis for a variety of severe infections that predispose otherwise healthy humans, infants, children, adolescents, and adults to a single type of infectious disease. His work tells us that life-threatening infectious diseases can be caused by what he calls inborn errors of immunity, or IEIs. We'll try not to use too many abbreviations today. Along with his co-author and long-term collaborator, Laurent Abel, Jean-Laurent has published a terrific article recently in the Annual Review of Pathology, Mechanisms of Disease, and it's entitled Lethal Infectious Diseases as Inborn Errors of Immunity, toward a synthesis of the germ and genetic theories. Jean-Laurent and I will discuss this work and how it relates to COVID-19. Now, do send your questions in the Q&A box at the bottom of the Zoom window under the YouTube video, or tweet them. So that's three different ways: Zoom video, YouTube video, or tweet under the hashtag #AR Pandemic Live. We have a team of trained curators who will be looking at your questions and feeding them to me after my conversation begins with Jean-Laurent. So with that, I think we'll start. Jean-Laurent, thank you so much for being here. Before we jump into COVID-19, which of course many of us are interested in, I think we should take a few minutes for you to give us some background on how and why genetics may play a role in the severity of infections. Is this a new idea? How long has it been around? What's the evidence for this kind of hypothesis?
Russ, thank you for inviting me. But if you ask me 3, 4, 5 questions in a row, I can't memorize them. I won't be able to answer them. Okay, so let's try to ask questions one by one. All right.
Okay. So
the
can
first
you give
thing,
us some?
Hold on. The first thing I want to say is to thank you. Okay. The second thing I want to say is that it's the first time I'm doing a webinar. Also the first time I'm on YouTube. So I'm thrilled and also to some extent intimidated or worried. Okay. Now, the third thing I want to say is to try to answer one of your many questions. And perhaps it's best that I introduce myself. I'm a professor of pediatrics, and my lab has been testing the idea that severe catastrophic infections striking previously healthy children, adolescents, or young adults in the course of primary infection may result in some cases, perhaps in many cases, from single gene inborn errors of immunity, that is monogenic disorders that are clinically silent until the patient is exposed to and infected with a particular microbe. Okay, so that's what we've been doing for 25 years.
How did you get interested in this line of research as worthy of your time?
Well, so I'm a pediatrician, and the most common problem faced by pediatrician is childhood fever, childhood infection, childhood death of fever and childhood death of infection. So this is really a fundamental problem in pediatrics. And what is really striking is that for every pathogen, virus, bacterial, fungus, or a parasite, children who suffer from severe clinical disease are actually infected by a microbe that is harmless in their relatives, sometimes their siblings, parents, also the healthcare workers. So the enigma that is faced by pediatricians and internists to some extent is that for any given microorganism, there is gigantic inter-individual variability in terms of the clinical manifestations of infection. It ranges from silent infection in asymptomatic individuals to life-threatening and sometimes lethal disease. What varies from microbe to microbe is the proportion of silent versus life-threatening disease. The proportions vary. Some microbes can kill less than 1 in 10 million infected individuals. These are very rare infections. These microbes are typically commensals or saprophytes. They're everywhere, and they're harmless in the vast majority of people, but in 1 in a million or 1 in 10 million, they can kill. And at the other end of the spectrum, you have viruses or other pathogens that can kill 1%, 10%, sometimes 20 or 30% of infected individuals. And in between, you have everything, microbes that can kill 1%, 0.1%, 1 in 10,000, 1 in 100,000. So this is this inter-individual clinical variability in the course of infection that I think is the fundamental infection enigma. And that's the problem that my lab has been interested in for now more than 25 years.
Great, thank you. So in your review article that I referenced in the introduction, you outlined 3 categories of inborn errors of immunity that contribute to the susceptibility for infection. Could you take a moment to review these 3 areas and perhaps give an example of what goes wrong or what is the genetic problem in each of these 3 areas?
Right. So the discovery that some genetic variations can underlie severe disease in the course of infection in a previously healthy individual, that discovery followed 3 sets of studies corresponding to 3 steps that I'm going to briefly try to explain to you. The first group of inborn errors of immunity are often referred to as primary immunodeficiencies. They were clinically described in the 50s, and the genetic variations, the mutations responsible for disease, were deciphered from 1985 onward. These patients typically miss a big component of host defense. They lack a subset of leukocyte or a molecule produced by leukocytes that is of great importance against a variety of infectious agents. Therefore, these patients early in life suffer from multiple recurrent infectious diseases, sometimes even often infectious diseases that are unusual, rare in the general population. A good example are babies who lack lymphocytes, such as the famous child who spent years in a sterile bubble to
the boy in the
him,
bubble.
Exactly, to prevent him from being exposed to environmental pathogens. So these are rare conditions. We said that they're Mendelian, meaning that they're caused by single gene lesions that are highly penetrant. That is, if you carry the at-risk genotype, then you will display both the immunological consequences, the lack of a leukocyte subset, for example, and the infectious consequences that themselves result from the immunological anomaly. So these are the primary immunodeficiencies, but you understand easily that this is very different clinically from, say, a cousin of yours who would have TB or a friend of yours who would have meningitis but would otherwise be normally resistant to infectious agents. But still, these primary immunodeficiencies provided proof of principle that in humans, infectious diseases could be genetic. Okay, so that is step one. The second step is a series of studies that have focused on what we call Mendelian infections. These are clinical conditions that were described earlier, for some of them, earlier than the primary immunodeficiencies, from 1946 onward. These are patients who are vulnerable to one microbe, only one. And this unusual vulnerability to one relatively rare infection segregates in families like a Mendelian trait. These infectious diseases are unusual because they're rare, because they run in families, literally, as a Mendelian trait. And there are different types of Mendelian traits, but they really run in families. And these patients are otherwise, paradoxically, normally resistant to other infectious agents, unlike patients with primary immunodeficiencies who, as we've discussed earlier, are prone to a variety of infections. So there are five such Mendelian infections. They were clinically described from 1946 onward, and from 1996 onward, the genes were cloned, and the molecular basis of these very unusual Mendelian infections were discovered. What's interesting with this small group of patients, and I'm going to give you one example, but what's interesting is that they provided proof of principle that infectious diseases striking otherwise healthy individuals, normally resistant to other infectious agents, could also be genetic. So that was the second step, getting closer to the situation with which you're familiar, your uncle or your friend suffering from a common life-threatening infectious disease. And I'm going to give you one example of these five Mendelian infections. It's a condition known as Mendelian susceptibility to microbial diseases. These children, adults, or children, adolescents, or adults are fine until they develop severe disease caused by mycobacteria, a group of bacteria that are ubiquitous in the environment, or until they're vaccinated with the BCG vaccine against TB, which is a live vaccine and that can also cause disease in these patients. Now, these patients suffer from inborn errors of gamma interferon immunity. That is, they carry mutations that reduce or abolish the production of gamma interferon or the response to gamma interferon. So what we've learned from this experiment of nature, so to speak, and from the other four Mendelian infections that we will not describe in detail today, is that there were truly genetic disorders that can make people vulnerable to one microbe. And with high penetrance, these are Mendelian disorders. If you carry the genetic lesion, you're prone to a particular infection. So that's step number two. The third step evolved from 2007 onward with the discovery that there were common infectious diseases in the general population that could be caused literally not only by the infectious agent, but also by monogenic lesions, inborn errors of immunity that do not display complete penetrance. That is, a child, an adolescent, or an adult with one severe infection, with no medical history, himself, herself, or in his or her family. That is, this infection is not running in the family. It's sporadic, it's isolated, and for a number of infectious diseases that we're going to discuss in a second, monogenic inborn errors of immunity have been discovered. So that is the third step. And the first, I think, discovery was published in 2007 with mutations in a pathway that underlie herpes simplex virus encephalitis. This is a relatively, I wouldn't say a common infection, but it is not a rare infection. It unfortunately strikes maybe 1 in 10,000 children. Herpes simplex virus is an almost ubiquitous virus. People are infected, they typically have gingivostomatitis, and when there are recurrences, they develop herpes labialis. But in, 1 in 10,000 children, the virus doesn't stop in what we call the trigeminal ganglia near, in front of the ear here. The virus unfortunately goes to the brain and causes encephalitis. Well, it was discovered from 2007 onward that this sporadic, tragic disease can be caused by inborn errors that impair immunity of neurons in the brain against herpes simplex encephalitis. So these are the 3 steps from 1985 onward that led from, the baby bubble, as you say, the severe primary immunodeficiency, kids vulnerable to everything, to the discovery of sporadic isolated infectious diseases of humankind being caused by monogenic inborn errors of immunity with incomplete penetrance.
Well, thank you very much. That's a beautiful setup now to give us a kind of a more deeper understanding of the setup for what you now know I'm going to ask, which is, what is your sense now of the situation with COVID-19 and the possibility that there are the same kinds of processes happening in the current pandemic?
Well, Russ, this is of course, an important question, a timely question also. Around 95% of the severe cases of COVID are probably accounted for by age or pre-existing comorbidities or both. It seems, whether in Asia, in Europe, in the Middle East, in the Americas, that most people with COVID severe pneumonia, people who require oxygen and admission to an intensive care unit, most of them are older than 50 or 60 or 70 years, and most of them have 1, 2, or 3 comorbidities, metabolic disorders, cardiovascular disorders, or pulmonary conditions. So that does not translate immediately into molecular, cellular, or immunological terms, but we understand from this epidemiological description that aging, sick, frail individuals are more vulnerable to SARS coronavirus than previously healthy young people. Now, in that context, it still is surprising and even shocking that 1 to 5% of the severe cases are younger than 50 years of age and do not display any overt medical condition. That can be someone who would be running the marathon in October 2019, and now in April 2020, he or she is in the intensive care unit, intubated and ventilated. So that's the problem that we're trying to tackle. And as you suggest, this problem is very similar to the problems that our lab and other labs have tackled in the last 30 years. Someone who would have severe flu for no reason, or someone who would have severe tuberculosis, and now, or severe herpes encephalitis, as we've discussed. And now it is someone with severe COVID for no apparent reason. So that's the problem we want to tackle. The way we want to tackle this problem, you want to ask a question. I'm sorry, I'm very long, I'm long and wordy.
No,
because
fact,
English is not my native language. In French, I would be much more concise, but I don't think you can learn French fast enough to interview me. Right?
Well, I can tell you I would be extremely concise if I were to try to speak French. But actually, I was going to ask, I think, the question you were about to answer, which is, you have tracked down in your career similar associations. So what kind of data needs to be collected in order for you and your colleagues to get started on understanding if this does in fact have the genetic influences that you suspect it may?
Right. So I've told you about the problem. 2 is the hypothesis, but you've just said the hypothesis. The hypothesis that severe COVID in previously healthy young people can be genetic, single gene inborn errors of immunity with incomplete penetrance. Okay, that's the hype. Now, you're asking me what's the third step, which is the approach. So because these are relatively rare patients, we decided with Helen Su at the NIH early on in February to try to collect patients, to recruit patients all over the world, and to team up with colleagues in Asia, Oceania, Africa, Europe, and the Americas. Why? Well, because what we've learned from the previous studies of infectious diseases is that there is what we call in our jargon genetic heterogeneity. That is, for a given infection, there can be mutations in different genes. So, for example, a patient with severe COVID in New York City may be mutated in gene A, and there's another patient mutated in gene A in Tokyo, Japan, and a third in Sydney, Australia. So the only way to suspect that gene A may be causative of severe COVID is to have these 3 patients, because then with 3 patients mutated in the same gene, the likelihood that the gene can be responsible for severe disease is much higher than if you have only 1 patient mutated in that gene. So we've decided early on to start an international consortium, which we've designated as the COVID Human Genetic Effort. And the website is, by the way, COVIDHumanGeneticEffort.com. And we are organized in a very simple manner. There are hundreds of hospitals worldwide that recruit and consent patients. They take a blood sample and DNA is extracted, and the DNA is sequenced by a sequencing hub that is regional. There are already, I think, more than 30 sequencing hubs in Japan, Australia, in the Middle East, in Europe, in the Americas. And these sequencing hubs analyze the data of the patients in their region, but also they work together so that all exomes and genomes from patients worldwide can be analyzed together in order, again, as I stressed earlier, to increase the chance of detecting genetic homogeneity.
So before I ask my next question, let me remind the listeners that if you have a question, you can post it on the Zoom, on the YouTube comments, or at the hashtag #ARPandemicLive. I know there are some questions coming in. We will get to those in about 6 minutes and 49 seconds. But my next question on that note is, this is a very exciting consortium that you've assembled, and we're all rooting for you to find out if we can give clinicians information about those who might be at greater risk. Is the consortium focusing on these young folks that you refer to, less than 50, no existing comorbidities, or is it a broader that's even including the older patients?
No, we really try to recruit young, previously healthy patients, no comorbidity, and younger than 50 years. Now, you may say rightly, well, 50 is arbitrary. What's the difference between someone age 49 and 51? True, there's no difference. And in addition, at a given age, people can be very different. People age in different ways. So it's a threshold that is convenient, but it's arbitrary. Therefore, if a clinician somewhere sees a patient age 60 or 70 who was in great shape and all of a sudden has severe COVID, that patient could be enrolled as well.
So as you know, I'm a little bit of a geneticist myself, and we always worry about the interaction between genetics and the environment. And so I'm wondering in this effort if there are plans either well underway or just still in the planning process to evaluate potential environmental factors in these young people that could also contribute to the severity of the infection.
Well, environmental variables may account for interpopulation differences. You can think, yeah, yes, maybe the epidemic will take a different turn in Ecuador and Iceland. That's plausible. But these differences are unlikely to account for inter-individual variability in a given region within Iceland or within Ecuador, for example.
Are there so that's very interesting, and I'm actually a little bit surprised by that. Because there is these possibilities that specific exposures in the diet or specific histories of other infectious disease in the past, which I would generally put under the category of environmental. It seems to me possible that those micro differences that could be different within a population, but obviously I haven't done this work, and it's interesting for me to hear that you believe that within a cohort, you with enough numbers you'll be able to average out those effects. I guess that's the argument.
No, Russ, I think we misunderstand each other. I misunderstood your question. So I'm not saying that the history of the individual does not matter. It may very well matter. As you rightly point out, the history of previous infections by other coronaviruses, for example, may be beneficial or detrimental when the host is infected by SARS coronavirus 2. That's unknown.
Right.
Okay. And I agree that, for example, the coronavirus SARS-1 that was limited to China, it may be that in Chinese people who had previously been infected with the SARS-CoV-1, maybe the course of SARS-CoV-2 infection is going to be better or worse. That's unknown, but it's possible. I'm with you there.
Good. Thank you. We're going to be going to questions from the audience in a moment, but I also wanted to ask, and I'm a little bit embarrassed to ask this question because I do research as well, and the research timescale is never satisfying to the public.
But don't ask me about the future. Don't ask me a question about the future.
I am compelled to ask you in the name of our people listening, could you give us any sense of timeline?
Yeah, let me look at my crystal ball. Yes, okay, give me a second. All right. Well, no, I got no answer.
Okay. I understand, and I accept that answer. So for those who are wondering about timelines, people work as hard as they can, but in science, you can't predict when the insights and when the observations come that are going to give you the answer. So I totally understand my colleagues' hesitance to answer anything about timeline. It sounds like they're working extremely hard, and that's really all we can ask.
Thank you, Russ. We're scientists. We're neither magicians nor politicians. Okay, yes. We're doing our best. We're working hard, but I can't predict the future.
Thank you. So we do have some questions, and I'm now going to go a little bit early because they look good. From Adam, we have the question, is your research showing potentially that only certain genetic mutations could predispose someone to a potentially lethal COVID-19 result?
I'm not sure. Did you ask me a question?
This is from Adam. Yeah, he's in the audience, and he wants to know, I guess, would your research discover a genetic mutation if it was strongly correlated with lethality of COVID?
Oh, yes. So, strongly correlated with lethality or severity of COVID would imply that we have 2 or more cases, probably dozens of cases, that is dozens of patients carrying mutations in a given gene. That is an optimistic scenario because it suggests that there is high genetic homogeneity, that we could find many patients carrying mutations in the same gene. It is possible.
And then Adam followed up with a much more specific question about whether it's possible that existing mutations that are known to predispose for other diseases like retinal blastoma or other kind of phenotypes, are they more likely to be the ones that we find, or is it possible that we're going to find something entirely new?
So retinal blastoma, it's hard to see a connection, but we have to be humble and agnostic. A more obvious example are the inborn errors that underlie pneumonia caused by related viruses, not coronaviruses, but influenza virus or rhinovirus.
Yes.
So there are already examples of viral pneumonia, severe viral pneumonia that is genetic. Now, these
And
inborn errors could also underlie severe COVID. That's possible.
yeah, so I think that, so in fact, Anna from the audience asked a question about whether it is possible that there are other specific viruses whose pattern of severity would correlate with the patterns of severity that we see with COVID.
Well, I think that the SARS coronavirus 2 is the most virulent virus since 1918 in terms of pneumonia. So there is no recent example.
Yes. There's a couple of questions. I'm sure you're familiar with biobanking efforts in the world, and in particular, the UK Biobank, which follows 500,000 residents of the UK and has both genetic information and some clinical information. They recently decided to release information about the subset of 500,000 patients. I'm sorry, the subset of 500,000 participants in the UK Biobank who have been infected by COVID. I'm wondering if that represents a useful, the question is from the audience, does that represent a useful resource for your consortium?
Yes, I think UK Biobank is an extraordinary gift to humankind. And this is really a wonderful resource. I wish there were more equivalent resources worldwide. And for our studies of tuberculosis in the past, we've used UK Biobank with success. And when they released the COVID information last week, since that day, we've been analyzing almost day and night the UK Biobank. So we're working on it.
Another question, interesting question, they're all interesting, is you mentioned that you're focusing on young people with no comorbidity because it seems to be a much more complex issue for the elderly. But this person asks, might there be genetic damage accruing over age throughout the lifetime of a elderly individual, damage to their DNA that actually does cause their increased susceptibility to the disease?
Yes, it is plausible. Another good example is zoster. Everybody or most people are infected with the varicella zoster virus in childhood. They have varicella, but then the virus is latent. But from the age of 50 years onward, zoster appears. And, you know, it's an indicator, unfortunately, of the inevitable aging of human organisms. And I think the same applies to COVID.
Well, somebody was listening very carefully to your useful summary of the three different categories of inborn deficiencies of immunity. And their question is it possible that this is a polygenic process in the sense that we don't find one gene, but it's the interaction between several genes that causes this increased susceptibility? And if so, would your consortium be able to detect that?
So the monogenic disorders that do not display complete penetrance and that have been discovered from 2007 onward, by definition, in fact, literally are not monogenic, because incomplete penetrance implies the involvement of other genes, perhaps a second locus, perhaps two, and perhaps more. So what we're searching is the main driver, the monogenic lesions, but we certainly do not exclude the possible contribution of a second hit or a third or a fourth, you know, but we start by trying to identify the key lesion, the key variation, the key genetic disorder.
So there's a very practical question. If a mutation is discovered through your consortium or for the efforts of others, can you imagine what kind of practical impact it would have for clinicians dealing with treatment and prevention? Or perhaps how has it affected it in the other examples previously?
Well, that's another great question. You know, I can answer in two ways. The first answer is a general answer. It is that the first step in medicine, in medical research, is to understand. Because once you understand a mechanism of disease, you can start thinking rationally about ways to prevent or treat it. Okay, that's a general abstract answer, but it's validated by 200 years of medical research. The second more specific answer is, as you suggest, based on previous examples. And here we're going to discuss, again, Mendelian susceptibility to mycobacterial diseases. These patients are vulnerable to mycobacteria because they don't make enough gamma interferon, or their cells do not respond well to gamma interferon. Well, it turned out that gamma interferon is a medicine that's been on the market for 40 years and that you can buy at the corner of the street. So the identification of the genetic and immunological basis of Mendelian susceptibility to mycobacterial disease immediately provided a way to prevent mycobacterial disease or treat it in these patients. You know, it's very similar to insulin for the diabetic patient. You essentially provide the component that the organism does not produce in sufficient amount. Now, I'm not saying that in 3 months we're going to have a miraculous component for COVID. I'm just saying that it's a rational approach to try to understand the mechanism of severe COVID, and that by analogy with other genetic disorders, that may at some stage lead to preventive and therapeutic advances.
Yes, and if I could add, as you know, people are now beginning to worry about how do we re-engage society. And obviously, if we had these very valuable biomarkers or indicators of who's at particular risk, we could come up with a plan where they get some special treatments, special management of their situation, because we now know if this information becomes available, that they would be at increased risk. So that really does make perfect sense as a public health as well as a personal health intervention. Now, we have many questions from people, this is actually very exciting, from people around the world. We have somebody in Lombardy, Italy, we have somebody from Brazil, and the questions are similar, which is, what are the chances in your assessment that there are country or geographically, geography of origin specific genetic mutations that are leading to higher rates in Italy or in Brazil or in any place in the world, versus that being a function of other non-genetic factors?
So that's yet another great question. You know, I hesitate to stick my neck out here, because if I follow my instinct, I would say that the inborn errors of immunity underlying the severe forms of COVID are probably equally distributed, you know, across ancestries. In the past, people would say across ethnicities. That would be my instinct, because I think that we're searching mostly for rare variations that are highly penetrant, and I would be inclined to think so. However, we may have surprises, and it may very well be that in some ancestries, there are more common variants that have a greater impact because they're enriched in that particular group of people, in that particular ancestry, or conversely depleted. So, you know, I'm not going to take any bet.
Yes. There was a kind of a related question that came up, which is, we've been talking about mostly about genetic variations that increase the risk of the disease. Somebody asked, is it possible that you will learn that there are certain genetic backgrounds that get protected from the disease?
Absolutely. Well, another fascinating question. It is not proven yet, but I think there's every reason to believe, and in fact to hope, that some individuals exposed to the virus again and again, for example, the spouse of someone who ends up in the intensive care unit, or healthcare workers with insufficient protection, would remain seronegative, that is, would not even be infected by the virus despite repeated exposure. These people, naturally resistant to the infection itself, if they are identified, would perhaps be genetically resistant. And there are examples for two viruses, the noroviruses that cause gastrointestinal lesions. Individuals homozygous for a variation in the gene called FUT2 are naturally resistant. The virus simply cannot enter the intestinal epithelial cells. Another well-known example is a mutation in CCR5. These individuals when carrying 2 copies that are mutated of CCR5 are naturally resistant to the human immunodeficiency virus. So there are already 2 examples, and there might be humans genetically resistant to the SARS coronavirus too. We're trying to detect these individuals, and if they exist, we'll try to find the genetic underpinnings.
The questions that are coming in are extremely excited about the consortium that you described, the international collaboration. It's very inspiring, I think. And one question, very simple, is how is it going with respect to sample collection? And are you at the hundreds or thousands, or where are we in that process? They just we're asking about the numbers.
So, I cannot speak on behalf of my friends and colleagues in Japan or Australia or elsewhere, but in Paris, we've already enrolled 100 patients, and we've already sequenced about 50. In New York, we've enrolled fewer patients, but it's also progressing.
Is it harder to get research done in New York? The reason I'm asking is we have lots of research happening at Stanford University. Our curve has been flattened pretty effectively. And so now we have this. It's not really a problem. It's good news. People are not as sick, but we also don't have as many patients as we thought we would to do clinical research. So there are some drug trials and some diagnostic and therapeutic trials that are actually stalling a little bit, because this is good news. I just want to stress that I am not complaining, but we're not having the patients that we were expecting. I think there might be an opposite problem in New York. And of course, Rockefeller University is in New York, where it was my impression that for the last few weeks, people have been so much just trying to get on top of helping these patients. I'm just wondering, has it been possible to get any research done in New York?
Well, yes, we've recruited patients, and we collaborate closely with university hospitals in the city. I think most, if not all, institutions in New York have teamed up with us, and with the New York Genome Center. Tom Magnetis, who's the current head of the New York Genome Center, has agreed to sequence the genome of these severe unexplained young cases for us. So they form one of the sequencing hubs.
And I think perhaps the final question before we close up is there's a lot of questions about whether you're looking at drug response and the genetics of drug response as part of this study.
No, we haven't so far decided to tackle this other important problem.
So listen, I think that this has been wonderful. I want to thank all of you for joining us. I want to thank Dr. Casanova for joining us. We hope you enjoyed it. We hope this was useful and science-based, because that really was our goal. You can find this and other episodes of Pandemic Live at www.annual, let me make sure I say this correctly, www.annualreviews, all one word, .org, where you can also find Jean Laurent's review article that we've referenced. And remember to join me on the Future of Everything on podcast or SiriusXM if you're interested. Dr. Casanova, thank you so much for your time and for your work.
Russ, thank you so much for your invitation.
This ends the formal portion of our program. I'm going to leave the Zoom.